Well, good afternoon, everyone. Today I have the pleasure to introduce you Shinya Ito. Shinya comes from the United States, from the Allen Institute, but he did a bachelor degree in Hokkaido University in Sapporo, in Japan, and then he was a postdoc in Indiana University with
grad student.
Excuse me?
Graduate student. Yeah. Yep.
But you work with John Bex.
Correct.
John Bex. And then you were postdoc at the University of California, Santa Cruz. Did you work in experiment there? Did you do experiments?
Yeah, I think I was also doing experiments in John Beck's lab.
Okay.
Yeah,
so was using characterizing visual and auditory stimuli using high density silicon proof for recording. He is a scientist level 3 in the Anton's Archipov group. Anton's is Javier's co-supervisor of the thesis. He develops a biologically realistic model, as he's going to show now, integrating electronic microscopic and electrophysiology derived connectivity with machine learning, optimization to reveal circuit structures, support computations, and advanced biological grounds in artificial intelligence. So thank you very much for being here. The floor is yours.
Thank you very much, Claudio, and thank you very much for inviting me for wonderful place. I'm really loving this island and also Spain in general. So my name is Shinya Ito. I'm as a described scientist 3 in Anton Archipos team. And today I'm going to talk about the deep learning assisted simulation of the cortical circuit. And here's the organization of the talk. I'm going to start with how we're going to collect the data from the brain at the Allen Institute, and next construction of the model circuit, how we train the network, and then describe what the trained network can do after and doing the data, how to do data analysis on those, and also why that matters for the future of the neuroscience and in general in science. So I heard that most of the audience are not neuroscientists, but working on complex systems. Okay. This is the pointer? Okay.
This or this to move the slides. No, you have to put it somewhere here.
I see. Does it work with let me try.
Hopefully it works.
Okay. I need to press allow button. And keyboard setup. Maybe I can quit this. It works. Yep. Yeah. So first, let me talk about collecting the data. And since this is not the neuroscience audio, I also inserted one of the slides for describing what the neurons are. And neurons are a type of cells in the brain. And this actually, modern neuroscience really started in Spain. And probably everyone knows Santiago Ramon y Cajal, who made a lot of the drawing with the so-called Golgi staining that is also developed by another, I think Golgi is an Italian guy. And the Cajal actually had drawn many of the beautiful drawing of the neuron like this. I think it looks like this is from the Cajal Institute at Madrid. And he actually is the first to develop one of the first people who developed the so-called neuron doctrine, which is a theory that the brain tissue works as consists of individual neurons that communicate each other. And that really started the modern neuroscience. And neurons are about 80% of the cells in the mouse brain. And it used to be called like something like 10% of all the brain cells. But this was actually corrected around 2010 by a Brazilian group. And now neurons are believed to be much more abundant. So it is actually the majority of the cells in the brain. Of course, like the density depends on the regions. Cerebral cortex slightly has larger density, or actually lower density of the neurons compared to the back of the brain, cerebellum. But it is the majority. And there are also a bunch of cell types and something like excitatory neurons that excites other neurons, inhibitory neurons that inhibit other neurons. And we are studying those cell types. And counting this number of cell types is actually not trivial. And it is also still ongoing study. I think we kind of believe that there are about 30 different neuron types in the mouse cortex now, and another, say, 20, 30 in other areas, and etc. So in total, there will be actually potentially hundreds of the cell types. And in order to study each of them, you need quite large scale recordings. If you just study individual cell types, like characterizing electrical property of one cell type at a time, you just need, say, 30 sets of experiments. But if you want to study the connectivity, you need 30 by 30 combinations. So the number explodes quite dramatically, and you need actually a great way of doing experiments. And that's where sort of the Allen Institute comes in. So Allen Institute is a institution that was founded in 2003 by Paul Allen, who is Paul and Jody Allen. Paul is a brother of Jody, and this guy is a co-founder of Microsoft, who built the Microsoft company with Bill Gates, and he actually had a lot of money and just created the institution for studying how the brain works, and called up many of the neuroscientists and other biologists and tried to determine what is one of the most important things to do in the neuroscience field. And a lot of the people gathered together, discussed, and decided we need a map of the brain first in order to even start understanding how the brain works. So the first mission of the Allen Institute was to create a comprehensive map of the gene expression of the mouse brain. And so, yeah, so the mission of the Allen Institute is tackling the problems that the individual labs cannot work on in a scale, and just to build up the, say, big pipelines for doing experiments and execute a lot of the experiments to produce the large-scale data set that everyone can use. And it started small. At the beginning in 2003, there were only 4 employees, but now it has more than 900 employees and 1 building plus a few different places as a workplace. Its mission is, I already partially stated, it's accelerating scientific discoveries. And at the forefront, just to read out the mission, at the forefront of bioscience, we're committed to understanding life and advancing health. We tackle big research projects through multidisciplinary teams and share our data and scientific resources openly to drive impact worldwide. So yeah, the keywords are big, open, and big, team, and open. So this is the kind of core mission of the Institute. So we do what individual labs cannot do to make everyone, in a way, happy for in data mining. So sorry, I forgot to say at the beginning, but please ask questions anytime. I'm happy to have an interactive session. So don't hesitate to ask questions. So here are some list of how we study how the brain works. I'm going to do explanation of each, but these are kind of 4 major experimental modalities. It's not all of them at the Allen Institute, but something that's related to the model construction that I'm doing. So it's only a part of it. PAT-seq is basically a patch clamp experiment. I'm going to also explain it later, plus the sequencing of the genes in order to make correspondence with the electric properties and gene expression of the neurons. And multi-patch electrophysiology is simultaneous patch clamp experiment in order to study how the neurons are connected. So if you just tap onto 2 neurons at the same time, you can see whether they're connected. And if they're connected, you can measure the electrical property of the information transmission between the neurons. And the third is extracellular electrophysiology and optical physiology. And instead of doing the experiments in vitro, which is the outside of the brain, you can also do experiments in vivo, which is the inside the living animal. And you can just put the either probe into the brain or measure the activity with a microscope in order to see how the neurons act in real behavior. So that's also another modality of experiments that we use for model construction. And lastly, electron microscopy connectomics is another major experiment that we're doing at the Allen Institute. This is a kind of very brute experiment. You basically get the 1 cubic millimeter of the tissue, slice them in a very fine tissue, and measure everything in electron microscopy and make a 3D construction of all of the neurons in there. So I'm going to do a little more detailed explanation for each of them. So patch-seq is basically measuring the brain tissue or one neuron and then put it into the sequencing. So the process is like this. So you have a neuron in the brain and you tap onto the neuron with the glass electrode. The tip of the glass is about like 1 micron diameter, and you just make by like elongating the glass pipette and bending it, and you can make the pipette, and you just put the pipette onto one of the neurons, and then you apply negative pressure. Then you can actually puncture the surface of the neuron, and you can infuse the dye which is used for staining the neuron later. And you can also measure electrical properties of the neurons inside. And you do some of the measurements in order to characterize each neuron. And then you're going to remove the nucleus of the neuron for later doing the sequencing. And it's going to actually seal off the where you pulled out the nucleus. So you can study the morphology, the shape of the neurons, electrophysiology, which is electrical property of the neurons. And then later you can do DNA, RNA sequencing of the neurons in order to get what kind of genes are activated in such neurons. So combining these 3 modalities, you can study single neuron properties in a detailed manner with all the information combined together. Yes? Yeah, thanks. So it is already like in the slice, and you infuse the dye in the neuron, and later you're going to just look at the neuron in the microscope. So everything is already stained, so you can just trace the neuron dye. Yep, and multi-patch electrophysiology is so the same patch clump experiment for measuring the electrical properties. And for this experiment, you also expressed some of the sensors for specific types of neurons. For example, for inhibitory neuron types, we measure properties of parvalbumin positive neuron, which is called PV neurons. I think they may show up later, like 3 major inhibitory types. And we distinguish everything and then measure the property for each one. And neurons also have, the cortex also has layers, like layer 1. In the mouse, layer 2 and 3 are combined together. So we call it layer 2, 3, and then layer 4, layer 5, and layer 6. So in order to study the probability of the connection between the neuron types, you actually have to make this kind of in this case, it's actually 17 by 17 matrix. It's like resolved into different layers and different cell subtypes. And then for each type, you measure connection probability by basically brute force. So they have done more than 2,000 experiments where they patch onto up to neurons simultaneously. I think this is actually requires very precise micro manipulator, and patching onto 8 is actually technically very challenging, but they've done actually a lot of experiments over 3 years. The inception of the project is 2013 and the completion is 2022. So basically they spent a majority of the time for building up the pipeline and refining the experiments. And in the last 3 years, actually not last 3 years, in the middle 3 years, they executed all the experiments and then spent also a few years after that for the data analysis and compilation of all the data sets and making also web interfaces, APIs for the later users. So that's the multi-patch experiments. It's one of the most important important data for characterizing connection probability, its spatial profile, and when they are connected, electrical transmission property, synaptic properties of those neurons. Yep, and the next one is extracellular electrophysiology plus imaging. So this is the so-called Allen Brain Observatory dataset. I think if you Google, you can also find the data are available. But the basic experiments are like you put the mouse on a dish and they can run on the disk freely. And then while they're doing some where they're exposed to some stimulation, you record the neurons either with the electrical probes that looks like this, or optical microscope. And depending on the measurement methods, you get either larger number of neurons. So optical recording has larger number of neurons, but it has lower time resolution because of the sampling rate and also the temporal profile of the calcium. It is actually measuring calcium flow into the neuron, which is actually something like a 30, 50 millisecond resolution. And for electrical measurement, instead you can't decipher the exact location of the neurons, you get the better temporal resolution. You can get the something like 50 microsecond time resolution for electrical measurements. Yep. So this is actually necessary for characterizing the neurons in action. And the last one is the electron microscopy. Oh, this was actually supposed to be a movie, but I'm not connected to the internet. Okay, so if you Google Micron's project, you can find a movie of the tissue. So this is actually already reconstructed tissue. Probably it's showing very tiny fraction of the neurons, something like 1% or less. I think it may be less than 1%. So tissue is basically filled up with all the neurons and other cell types. And but you can get the very high resolution of each neuron after reconstructing those things. Raw data size is about 1.6 petabytes. It is the 1 millimeter by 1 millimeter by 1.25 millimeter. Resolution is, you slice this into 50 nanometer sections and resolve each image with the 5 nanometers. So this is going to be 25,000 slices with the 200,000 by 200,000 pixels. If you compare that with the 4K TV you may have, this is going to be something like 5,000 times more for each image. And the problem for this experiment is if you miss one slice or mess up one slice while slicing, you have to do another experiment. So this is going to be very, very challenging experiment. And they also refine the rig for spending a very long time and completed the slicing methods for doing these experiments. And this was actually a very large data set and resulted in something like 7. So yeah, they talked with the Nature editor and published a collection of articles in Nature, which resulted in 7 Nature articles, 1 Nature Neuroscience, 1 Nature Method, and 1 Nature Communication paper, which was published early this year. Yep. So now using these data, my job is to construct models. So why do we do modeling? So I think the motivation for modeling is making a kind of virtuous cycle between experiments and theory or modeling. So we get a lot of the information from the experimental side, and we make some model, like either biophysically detailed neurons, which includes morphology of the neurons, or approximate each neuron by just a point to make point neuron networks. But in order to run such a model, you need to identify and decide all of the parameters in order to have the model running. So that clarifies the where in the experiments the data are lacking. And that's also one information we can give back to the experiments. And also it can do a lot of the predictions, since experiments are never kind of complete experiments. And where something is not measured, you can predict by running those simulations and give information back to the experimentalists. And if there are any interesting phenomena or non-trivial phenomena, we can talk to the experimentalists to check those things. And then we can make a cycle for expanding our knowledge with those cycles. And so those very detailed models are also important for making a little more simplified modeling work by just say, if you have, say, 10,000 neural model with a detailed morphology and parameter sets, you can simplify those models for a starting point of simpler modeling work. So this might be actually useful for either experimentalists and the modelers as well. And we also already have one published model in 2020, which is the Spiller et al. Paper. I think Javier would talk about this in his thesis in 2 days, right? And so I'm gonna spend just a brief time for explaining it. So this is actually the first model, first full all-layer visual cortex model that our group has published. And to briefly summarize what the visual system is, there's an eye and there's a retina in the back of the eye that sends information to a structure called lateral geniculate nucleus, which is in the thalamus. And this structure sends information to the primary visual cortex. And then the primary visual cortex sends information to the rest of the cortical areas. So our model is right on this primary visual cortex, and its radius is about 850 microns. And there are two versions. One version has the biophysical neurons and surrounding leaky integratifier simpler neural models. And the other model has all the neurons made in simple version. Why we made two versions is actually simply computational reason. This biophysical model that simulate all of the compartments of the neurons is computationally very expensive. You need to solve the cable equation for every part of the morphology, and basically spends about 10,000 times more computational resources just to run this model. So we sometimes need the simplified version of the model. But still, this one requires something like 3 minutes to run 1 simulated second. So this is much slower than the real-time simulation. So yeah, so if you start simulating like hundreds of thousands of neurons, it's going to be a little laborious for the computers. So based on this, we're actually developing a newer version of the model. And since then, so when we published this model, the two of the data sets were not published yet. So one is the multi-path synaptic physiology data set and the electron microscopy connectomic data set. Those are actually necessary for characterizing a lot of the connection information. But since they were not available last time the model, that was largely relying on the survey of the literature, which was actually very difficult to use because of the inconsistent information and inconsistent experiments that each lab has done. And those experiments are not really purposed for surveying the connection, but the more oriented towards like proving their own theory. So so we actually needed those data sets. So the first constituents of the model is cellular information, and that was actually published before the last model, which is kind of realized by cell types database at the Allen Institute. So we just also use that model. We use that database to create the model. And for structural information and connectivity information, we incorporated those new data set, multi-path synaptic physiology and the electron microscopy connectomic data set. So that provides the rules of the connections, connection weight distributions, and synaptic properties. So I'm going to go into a bit of the details of those things. So, for example, this is the one snapshot of the microns data set. And these images are really detailed, and you can even see like this wiggling structure of the V1 neuron and the axons coming from the LGN and where they touch, exactly like where they touch each other and make synapses. And this is actually not like imagery or anything, but this is a raw data in the microns data set. And this is only showing a few neurons, but you see you have this for every single neuron, which is, I think it is also hundreds of thousands of neurons in the microns data set. So this is, you can see, kind of appreciate the resolution of that data set. And from this data set, we reconstruct the axons, which is the projection kind of hand from the LGN that touches the V1 neurons, and those things. And we can calculate the connection probability for target populations and the spatial distribution around the neurons, like how it decays over the distance and whether they have like any skewness over the different angles. So those things can be characterized, and also the number of distribution of the synapse numbers. And it turned out to be actually close to the power law distribution if you measure those things. So you can characterize those things. And for recurrent connections in V1, we also have all the information at the resolution of the cell type pair. So in this case, it is 19 by 19. And connection probabilities is actually measured not as a single number of probability, but it is actually modeled as a function of the distance between neurons. And also in the actual synaptic physiology data set, I think a few of the corrections for experimental factors, something like signal to noise ratio also matters for detecting the connectivity and sorry, what's the other thing? So yeah, distance from the surface of the tissue also kind of matters because if you measure the neurons near the surface, those are more likely to be amputated during the slicing. So those things are also corrected, and we can get the connection probability for each of them with all those corrections. And not just the connection probability. If you go to the EM data set, you can measure you can actually fit the number of synapses on each neuron and characterize that as a log normal distribution. For physicists, I think those fitting might not really qualify for a lot of the details, but I think in biology, I think being able to fit as a specific function is already a great achievement. So please forgive without that we're not doing proper, say, chi-square tests and Monte Carlo rejections. But I think that those are majorly characterized as a log normal distribution, and we can actually get the three of the parameters for characterizing log normal distribution for each of the cell type pair. Sorry, so this is actually just a cell type information. Sorry. And for synaptic weight distribution, we are doing proper log normal distribution for each cell type pair where the data are available. But it also has some of the parts where the data are scarce. In those cases, we have to sometimes combine the information with the different cell types, but we try to do analyze at the limit of the data scarceness versus the reliability of the fitting. So that's the connection probability and making the distribution of the degrees. And for other things, the synaptic properties is also another important factor. So when they're connected, they transmit the chemical information around the neuron, and chemical information basically cause a reaction of the postsynaptic neuron to cause some influx of the ions. And those influx of the ions is going to basically become a kind of stimulator for the postsynaptic neurons, which may or may not lead to its own action potential. But this is one example of those chemical reaction in the postsynaptic neuron, and this is called postsynaptic current function. It's a function of time, and you see this temporal profile for each neuron pair. And there's also an improvement from the previous model. Previous model didn't resolve this in the different cell type pairs, but new model does it and also estimate this function in a better way. Previously, we were using something called alpha function, which is a one parameter function, basically have linear onset plus exponential decay. But now we prepare a double alpha function, which is addition of two alpha functions that fits the experimental data much better. So it is actually suggesting that this synaptic transmission may not be through just a single type of channel, but it is actually through multiple types of ion channels, and it is causing fast rise time plus slower decay time using those two channels. And it also indicated that there are interesting cell type differences for the synaptic transmission. So this is the table for transmission of one of the parameters. This is rise time of the synaptic time constants, rise time constants for synaptic properties. And I think, the labels are messed up. The source is actually on the y-axis, target is on the x-axis. So this is indicating that the one inhibitory cell type, PV neurons, whenever they receive synaptic inputs, the communication of the synaptic information is actually quite fast. That goes into PV neurons. And for example, like if this one is if the SSD neuron is the source, they actually transmit the synaptic information rather slowly, and they actually have almost factor of 3 difference in the temporal time constant. So you can see that appreciate that the different types of neurons actually use quite different transmission method in order to communicate information. So here's a brief comparison with the previous model and a new model. Compared to the new model, compared to the old model, the new model has slightly smaller diameter. I think this is actually the reason. The reason is since we actually have to put everything into a limited amount of GPU memory for the later training, we actually had to limit the size a little bit. So I think currently the maximum size that fits on one GPU is 700 micron diameter if the memory is 40 gigabytes, which is for a 100 NVIDIA GPU. If you have a bigger GPU memory, you can actually have more neurons. But anyways, I think our workhorse model is actually 400 microns, which is about 66,000 neurons. So this is sizably smaller than the previous model, but this is actually fine for characterizing a lot of the features. And compared to the last model, we also have large number of cell models thanks to the expansion of the cell types database since the last construction. And the number of LGN connections, thanks to the EM data, we could actually expand the number of, well, we kind of realized that there are a lot more LGN synapses in the V1 than the last iteration. So we increased the number, and the number of recurrent connections also mildly increased. So number of receptor types, this is basically just not the ion receptors, but the connection types from that I just described in the previous slide. So instead of using just the 4 synapse types, we are using 91 different synapse types. And again, the distribution of the in degree used to be Gaussian, but we now have a proper log normal distribution for in degree. And the weight distribution was also uniform, but we also now made it log normal distribution. So this is the kind of schematics for the new model. I think there are different cell densities in each layer. So this like one green circle describes the border between layers, and those red dots are the cells. And this blue lines are connection from one LGN neurons to whatever connected in the V1. And this yellow one is second degree connection from this LGN neuron. So showing the connection from whatever that this one particular LGN neuron connects, and from those neurons to all the other neurons. So by just going 2 degrees from a single neuron in the LGN, you get to pretty much the whole network. And there are also, I think it is tens of thousands, sorry, it's 8,000 times. Yeah, I think it is actually more than 1 million connections just by second degree from one LGN neurons. So it is the level of complexity of the network. Any question? Yeah.
This information comes from the electron microscopy.
So it is, yeah, both electron microscopy and the synaptic physiology. So yeah, connection probability can be extracted. So there was some like a history there. So we originally using probability from multipath synaptic physiology, and then later we also confirmed the connection probability is more reliably calculated in the electron microscopy, and cross-check those things for the consistency and switched to microns, electron microscopy.
And like when you do this slicing with this specific height, this specific sizes, can you reach LGN? How do you know that an action which reaches to a specific site is from LGN?
Like instead of other thalamic areas? Yeah, so it is kind of assumption. So most of the axons that exist in the V1 is actually from LGN. So yeah, I think you can exclude the other axons. There might be some small number of other axons that's coming from thalamus, but basically the methods are trace the, you can't trace all the axons back to LGN. LGN is not in the parts of the data, but you can see they're coming from thalamic pathway. So we know for sure they're coming from thalamus. And if they're coming from the thalamus, we just assume that those are LGN axons. Yeah. Yep. Any other questions before getting to the next section? Yes.
But from data, you see what is the distribution of the connections, and then you generate connections with this sort of distribution, or
Yeah.
are you cloning what you see in the experimental connections?
The former. Yeah. I think in principle, it is possible to clone those electron microscopy, but there are actually a few problems. So electron microscopy requires something called proofreading, since the axons are very thin and there's no 100% reliable automated method for tracing all the axons. So it is actually still manually done. So humans are coming in, looking at one axon and trace towards all of the, like in the whole the arborization. And that is called proofreading. And there are only about 1,000 neurons that are fully proofread. So you can only get, say, about 1,000 neurons that are reliably identify all the connections. And in principle, you can kind of fill in the rest of the information to create model, but we took the kind of more kind of statistical route, like measure just the statistical properties. This is also kind of primitive things, like we just are getting basically first order or pair-wise connectivity information, not even like triplets, and measuring the connectivity and reproducing statistically those connections. Does that answer your question? Yep. Go ahead. Yeah. They're working on it. Yeah. There are like a. Yeah, very good point. So I think this is actually an older project than ChatGPT. So those language models weren't available when they started, but there are also like a lot of the machine learning efforts for doing some automated proofreadings. And those are still within the pipeline. They're not as reliable as human proofreading yet. But with enough data but with the human proofreading, I think everyone believes that's the future. Yeah.
The only direct data you got from the imaging was the positional data from the neurons, and then the rest is basically statistical properties inferred from the externals, right?
So sorry.
The only direct data you got from the imaging is the positional data from the neurons. Or what's
the electron microscopy? It is so all the position of the older neurons plus all the arborization of the all the neurons as well. So yeah, all the axons are only for 1,000 proofread cells, but the dendrites are more reliably automated proofreading. So yeah, you can identify basically all the incoming synapses for each neuron, but outgoing synapses are more difficult. Yeah. So this is kind of for those who work on the modeling. So yeah, so everything what I told about is basically parameter extraction from the model. And if you just use those parameters, the amount of data that we are using for modeling is actually quite small. It is only like 1.2 megabytes, including the code for generating those analysis and all the parameters. So my GitHub repository for making the model is only 1.2 megabytes. If you download it and run one command after installing all the dependencies, you can actually download some information of the cell types from Allen cell type database, and it's going to run all the scripts for generating the model. So you can actually recreate the model in a single command, and you can also intervene at any step and modify for your own purposes. Like this is the visual cortex model, but if you want to model other areas, you just swap some of the parameters and rerun this pipeline for redoing those things. And it's going to also generate some of the run the simulation, do some primitive analysis. So I think everything is in one pipeline. So this is going to be a more efficient and reproducible and a transparent pipeline that we are creating this time. So the next section is training a network. This is an interesting part. And I assume also Javier will talk about some of the details for this section. So I'm going to try to deliver you only some limited information here. I think the story, starting of the story is this paper that was published from Wolfgang Ma's lab in 2022. And this Chen et al. Paper basically picked up our 2020 model, the model we published, and then they converted this into differentiable model, and then rewrite the simulator in TensorFlow, and then do a made this biological simulation available with the training. And this is the Chen et al. Paper implementation, and they published the paper in 2022. And it was actually quite impressive work, and we formed a collaboration with the Wolfgang's Ma's lab and the Claudio's group. And Javier and I spent 1 or 2 years for tuning the model. So we took the code from this Guozhan Chen's implementation, but it was not able to run the new model because of the added complexity of the new model. So something like the receptor type, number of receptor types were also a big hurdle for running this like TensorFlow on GPUs. So we have to do some of the engineering for reducing the memory use and making the computation also faster. So Javier basically did I would say 80%, 90% of those tuning works. And I also gave some idea or provided some ideas and had long discussion for many weeks. And then we actually got a differentiable version of the new V1 model. So probably you're going to hear the detailed story from Javier's defense. So without the tuning, we actually have not correct firing rates or nor correct properties of those networks. So this is the so-called raster plots. Each dot is expressing spiking activity of the individual neurons. And these are something like about 16,000 neurons at the core of the neuron. Different colors indicate different cell types. The majority is those red excitatory neurons. And this is also exposed to visual stimulus, so-called drift and grading stimulus. This is a kind of stripe that's moving towards a particular direction. And this is the onset of the stimulus. And you see kind of subtle increase of the firing rate or number of spikes from here to the later time. And this is sort of the baseline without any training. And after that, we're going to do adjustment of the firing rates. And we are kind of successfully adjust the firing rates, but this rate adjusted model actually didn't have the right properties. So some of the interesting properties of the visual cortex is orientation selectivity or direction selectivity to the moving directions. So some of the neurons specifically fire towards, say, a particular direction, and some other neurons like the other directions. And those are called either orientation selectivity or direction selectivity, depending on whether they care about the exact direction or just the orientation of the stimulus. So orientation selective neurons like to fire both this direction and the opposite direction, and direction selective neurons only fire to one direction. And you can also measure how, what degree of they orient, what strength, how strongly they are orientation direction selective, and this is what it's measuring. So gray is the experimental data, neuropixels, and orange is the baseline model, this one, and blue is the rate adjusted model, and this is the trained network with the back propagation. And if you just not train or just the rate adjusted firing rates, then you're not going to get the correct value of the orientation selectivity. And only if you train the network with a proper objective function that I would explain in the next slide, you can get all of the properties correct. So for example, baseline doesn't have right firing rate, rate adjusted model doesn't have good OSI, and still baseline models have reasonable OSI or DSI, but in the deeper layer, they also have some trouble. And this green trained network resolves all of the measures. And for achieving those activities, we use the objective function for training the network, and that is consisted of firing rate loss. Basically, it matches the distribution of each of the 19 different cell types firing rates. So fire rate is also measured in a neural pixels recording, which measures the activity of all the neurons, and you can get the distribution of the firing rates for each cell type, and we're trying to match that. And another term is voltage loss. This is a penalization of the membrane potential that goes below the resting potential or above threshold. So basically keeping the voltage level in a kind of reasonable range that neurons typically have. And this OSI, DSI loss, orientation selectivity index or direction selectivity index, this is a single trial population level OSI, DSI. And we're going to match also the value with the experimental data. And we also measure Fano factors for each neuron that basically shows the how Poisson-like the activity is. Like if the neurons are basically just a random process, it's going to have Fano factor of 1. It's Poisson process over time. But neurons typically expresses like large values of Fano factors. So we measure those things with the various spin sizes and try to match with experiments. And the last thing is the synaptic weight loss. So we measure synaptic weight distribution from the multi-path synaptic physiology data set. And in order to preserve those distributions that are originally set in the model, we're going to use this loss function for keeping the distribution for each of the cell type pairs. So if you use those functions, you can get all of the good properties. And then this is showing the progression of the H loss function, and total loss function actually goes down very nicely with the training. And here's the sort of the kind of showing the progression of the training, starting with the kind of low firing rate at the beginning, and it's going to develop the firing rate for upper layers first, and then layer 6 at the bottom layer starts to grow the firing rates and start showing this kind of orientation selectivity, which shows kind of clustered behavior over kind of this vertical axis, which shows which is sorted by actually preference of the orientation. So this clustering here indicates the neurons have actually preferred angles. Yep. So I think one big things to take here is the difference of the use of the activity data. So activity data were collected for a long time, but they were only used for kind of verifying the model construction. So previously, we basically manually tuned some of the parameters and basically pray that the activity matches with the activity. But with the backpropagation training, we can now use those activity measures as sort of target objective function and propagate that information to the structure to actively change the structure of the network to realize some of the like interesting structure of the network. So that's the major difference that we are doing in this iteration. So 50 minutes. I'm gonna try to go quick from here. So yeah, and the manual optimization also takes very long time. Like previously, each simulation was used to take like a few hours to do one iteration. And if you want to do optimization manually with that, and the number of parameters is like tens of parameters, then it's going to take actually months to just do the optimization. And backpropagation also resolved that problem. Simulation also runs a lot faster on a GPU. It's almost close to real time. So a few hours to a few seconds is one big improvement. And also automated training method over tens of millions of parameters, another improvement. So that basically resolved a lot of the issues. Last model, I heard that Anton spent 9 months to just optimize the network, which is nightmare. But now we can do it in half a day. So let me just briefly mention what it is. So there are actually so now I'm going to talk about what the network can do after training, or what the network properties are like after training. So this is something measurement called most exciting inputs, which is basically an image that excites the neurons the best, that is optimized for each neuron. So this is an experiment that done in Andreas Stolia's lab in 2018. What they did is they show a bunch of natural images to the animal, and animal sees those things, and you measure the neuronal activity by microscope, and And they also tried to make a convolutional neural network version of those neurons by fitting the data to those activity patterns. And then they get sort of like a digital twin of the individual neurons in the experiments. And once they get the digital version of those things, they can use diffusion-like methods to generate an image that excites those digital twin the most. I think in the experiment, they also play those movies or those images back to the real animal and verify this is actually more exciting than any of the stimuli. But we also did the same, used the same pipeline and replaced this mouse brain part with the model, and then tried to do the same pipeline and try to characterize this so-called MEIs from the real neurons versus model neurons. So here's what I found. These are the images, most exciting inputs for some of the neurons that they found in the real mouse brain. And here are what we found in our model. So admittedly, I also cherry-picked some of the neurons that look similar to those, but it is actually kind of easy to find such neurons that have similar properties as the real neurons. So I think this kind of one verification that we are actually doing quite well with reproducing what the neurons are responding to. And they only did it for upper layer excitatory neurons, but we can also do for deeper layer excitatory neurons, which have a little more complicated properties, and then for inhibitory neurons, which also has further properties. And if you measure, for example, like a distribution of the spatial frequency of those neurons and compare the high versus low spatial frequency component, upper layers have relatively low spatial frequencies, and deeper layers have relatively high spatial frequencies, which is indicating that they are processing a little more complex spatial patterns. And the inhibitory neurons will have similarly high spatial complexity. Yes?
What that layers have the largest values of radiation sensitivity. Doesn't that translate to the kind of maximum exciting images that they form? Like, should you see more stripe-like patterns for layer 6 excitatory neurons?
Yeah, that's an interesting observation. We didn't really see striped patterns in the deeper layers, and I'm not exactly sure why. Yeah. So in principle, like as Javier said, layer 6 has the strongest orientation selectivity, and if they're strongly orientation selective, you're going to have those like stripe patterns in those layer 6 neurons. But at the same time, layer 6 neurons also have more interesting response patterns. Sorry, it's going to be a while back. So yeah, compared to the upper layer, which responds to each phase of the stimulus, like this is actually a 2 Hz stimulus, and this like one clumping of the activity here is indicating that responses to one phase of the drifting gratings, like one black line crossing the neuronal groups. Layer 6 actually has less of them, like this is more like continuous firing. So instead of firing at the exact location, they're actually firing at the more like abstract information of the moving stripes. So if you try to extract the exact spatial pattern, it's not really localized. I think that might be one potential explanation. Sorry, okay, so I'm gonna skip to this slide. So now one important distinction from the previous iteration of modeling effort is that we're actively changing the synaptic weight of the created network by training. So you can actually try to see if there's an interesting structure emerged by those trainings. There's also so-called like-to-like rule known in the field, which is the correlation between the connection patterns in the network versus the response similarity of the neurons. So if you take like 2 neurons and check whether they respond to similar patterns, and if they have similar responses, they're more likely to be connected or more likely to be strongly connected. So this is showing some of the patterns. So this is showing a difference of the preferred direction of the neurons. And if you try to plot it on the x-axis and try to measure the synaptic weight or over the y-axis, for example, for excitatory to excitatory neurons, if the difference is close to 0, they have stronger synaptic weights. Originally, it starts with a flat distribution, But after training, you start seeing those structure showing a correlation of the strength of the connection versus the property of the neurons. And that's the case also for excitatory connections. And for inhibitory connections, interestingly, you see kind of anti-like-to-like role, which is the opposite of the like-to-like role. So they have actually the stronger connection if the preferred direction is orthogonal to each other, and weaker connection for the same direction. And I also have some like experimental verification of those properties, but I'm gonna just briefly mention that this is the real experimental data for those things. And the distribution actually looks kind of similar. This is collected from the electron microscopy data, which is also combined with the activity measurements. So, and this is some like Monte Carlo measurement for showing the statistical power of those analyses, which actually nicely agreed with the experiments. I'm gonna skip the details. Sorry. So I've demonstrated the workflow for building biorealistic network from complex data that the institute is collecting, and the method for training with the backpropagation. Please listen to Javier's talk about the details of those, and some of the methods of analysis for checking consistency with the literature. And this actually is a large integration of the transcriptomics, the gene analysis, and single neuron, multi-neuron physiology, ultrastructural connectivity data, and neuropixels recordings in a single modeling framework. So this is the big achievement of the work, and the resulting network exhibits realistic activity and allows investigation of the mechanisms. So I think I want to talk about also why this matters, but this is a bit over time. Is that okay to have 5 more minutes? Okay. Yeah. So why does this matter? I think eventually this might be connected to the understanding of the artificial intelligence. What we made is basically a neural network that can be trained with the same method as deep learning. So we can actually bridge some gap between how the real brain works versus how artificial brains work. And if we solve that such things, we might be able to solve the energy consumption issue of the artificial intelligence. So it is recently NVIDIA and OpenAI tagged together to create the 10 gigawatt data center. If you put in the framework, this is going to be 20 million high-end consumer GPUs, or actually total power demand of 20 Mallorca islands. Just look it up, the energy consumption, not just homes, but all the electricity in the islands. So that's a lot of energy. And potentially this can be solved by making a neuromorphic computers and using, instead of those GPUs, if you use neuromorphic computings, maybe that could be solved. But neural computing computers require spiking neural network. And what we currently don't have is good algorithms for spiking neural network that performs as good as non-spiking artificial neural networks. And this is actually necessary for enabling large language model to work on neuromorphic computers. And another thing is the local learning rules. So this is actually just for inference, but another thing is local learning rules that work as well as the backpropagation. That's also another information that we are lacking. And if we get that, we can train more language model on the neuromorphic chips and then resolve the problem of the energy. And what we can offer is the laboratory for testing hypotheses for making neural networks more efficient. So this is basically by careful interpolation between those biorealistic networks and artificial networks, what works well on the biorealistic networks, and determining how to port in those information into the large language model. And this is going to be also another thing is opportunity for learning more about the nervous system. Now we can learn from artificial neural network and try to get inside of the neuronal computation and try to understand in the model and eventually in the experiment how the brain works better. And this is the actually emerging field called neural AI. And I want to advertise. We're actually doing a first workshop of neural AI at the Triple AI conference. So if you have work around that area, please submit the paper think, sorry, deadline is actually approaching, but it's still available. So please consider submitting. And another thing is we are also making a software for making such modeling, large-scale model, and that's called Brain Modeling Toolkit. And we also have an annual workshop for learning how to use Brain Modeling Toolkit. There's also a small amount of travel support, so please also consider applying for the workshop as well as subscribing our newsletter. We're going to only send a few emails per year, so it's not going to be annoying. So if you just Google BMTK, you can find such information. And lastly, so sorry, over time, but we're also calling for Shanahan Foundation Fellowship. This is a program that brings in computational people into neuroscience. You don't need neuroscience experiences for applying for this. The deadline is December the 1st, and you need a PhD for applying for this. And if you recently got PhD, you can apply for this opportunity and study at Allen Institute for 3 years with salaries, and you can actually collaborate anyone at the institute and also University of Washington for doing your research for 3 years for training. So please think about it. And I also like to thank a lot of the people who are involved in this, including my own team, Anton, and also, of course, Wolfgang and Claudio and all of the people in the experimental teams. Sorry. And thank you for your attention.
Thank you, Shinya, very much. Do you have any question? You made already some, but any other, Emilio? I see you trying to.
Okay, this is the Allen Institute. There is also an Allen, there is also an Allen Institute for Artificial Intelligence, isn't it? Also in Seattle.
Yeah.
So do you have connections with them, or?
We have weak connections with them. So the AI, so that's AI called AI2, Allen Institute for Artificial Intelligence. And we are south of the Lake Union. They're north of the Lake Union. So we are kind of on the other side of the lake. I think they're also playing soccer occasionally, but not too much academic communication. But it is also a growing field. So now we have another section for doing artificial intelligence at the Allen Institute, which is communicating more with the AI2. So the communication is kind of becoming stronger. But there are two independent research institutions, although both are funded by Paul Allen.
I have a short question. Some years ago, we started to work with optimization function also to fix data, to fix parameter to data. And it was very simple when you use synthetic data. So you generate from a model and then you use that parameters with another initial condition for the model to fit it, and it worked very well. But it didn't work that well in experiment. Maybe because the model was not good enough for the neurons we had in the tissue. That was slides. So do you think that when you fix better the parameter is because the connectivity dominates over the dynamics modeling or whatever you want to call of the cell itself?
Sorry, I don't think I caught all the context. So you're doing, what are the synthetic data and what are those activity data?
We had synthetic data to fit, we fit the model with synthetic data, and we tried to do the same with experimental data with some model. And it didn't work very well at that time. My question is, could that be because of the model we used was not good enough for the real neurons? And do you think that when it works is because the model is not that relevant for the dynamics and it's more relevant probably the interaction between the neurons?
Yeah, a few things can be considered. So of course, in the synthetic data, you basically get only like what you put into the synthetic data. So I think it's going to have maybe lower dimensions. In the experimental data, if it's in vivo data with the active animal or awake animal, there are a lot of inputs from other brain regions, and also awake versus sleeping, running versus stationary, and those dimensions might not be covered in your model. So I think it was maybe difficult to fit those dimensions that are not really embedded in your model side. So you may need some more. Yeah, I think if you have some like latent dynamic parameters that can modulate your model, that might fit better.
Yes, Sandro.
I mean, you used backpropagation, right, for the training.
Yeah.
What what was the factor? What made it unviable to use other types of methods? I don't know, like evolutionary methods or other types of more biologically plausible, perhaps, than backpropagation.
Yeah, like 3-factor learning. So, sorry, is your question whether they can be considered?
Whether, why not?
Why not? Okay. So the reason why we used the backpropagation was since that was already developed and known that works well for fitting the functions. Definitely, we can consider using other methods. So, for example, like evolutionary algorithm was used for some other efforts, not this particular work, but for example, fitting biophysical neurons to the data. And I think they're kind of complementary. I think if you combine the backpropagation with the gradient descent plus evolutionary algorithm, I think it actually works better than the single one of them. So I think we can also try to consider combining them in the future with this kind of large scale model plus say evolutionary algorithm and running all of them in parallel. That's one way. And the local learning rules is also a developing field, and I think that's something we're going to appreciate a lot. Currently, basically, we just haven't tested them yet. We didn't have a bandwidth for testing it yet. And there's a very big potential that that's going to work as well as the backpropagation depending on the problem. For simpler problem, I think it is known that they work as well. And for complex problems, we don't know yet. But I think we are also interested in trying that.
Well, okay. I think it's Shinya will be here until Thursday afternoon because he's part of the jury of Javier's thesis. So, Shinya, thank you very much for being here, and thanks again.
Thank you very much. Yeah.